Welcome back to plant developmental biology. We are continuing shoot development and particularly organogenesis during shoot development. So, in previous class we have discussed the coordination between shoot apical meristem maintenance as well as the organogenesis in the peripheral region. In this class we are going to take example of leaf development. You know that during vegetative growth the major organs which plants make is leaves, and there is a huge variations in the morphology of the leaves across the plant. So, you can have different plants having a different shape, different size, different arrangements of the leaves. So, for example, if you look maize, Arabidopsis, rose, there is a huge variety in the morphology of leaves. But if you look the pattern of their development, so there is a quite similarity. Of course, there must be some species specific differences, but what happens generally during the leaf development, as you have seen in the previous class, that the meristem is being maintained here in the center, and then in the peripheral region the pramodia is getting positioned. So, in case of leaf development, the pramodia is going to be leaf pramodia, and this is positioned by generating auxin maxima in the peripheral region, and the one auxin maxima is generated here, the next auxin maxima will be generated at a particular distance from the existing leaf pramodia, and then like that you see a pramodia patterning and differentiation. So, once the pramodia is positioned, the pramodia will start the program which is leaf specific program, and then during the differentiation it will make all the tissues which are specific for the leaf. It will ensure the polarity in the leaf which is adaxial and abaxial, and the proximal and distal polarity. So, how it happens? This is a kind of typical picture of tomato shoot. You can see that this is meristem, this is leaf pramodia 1, P2, P3, and then if you see the region, these are the morphogenesis. These regions are basically differentiating, and eventually they are giving mature leaf where you have this different part of the leaf. This is a leaf morphology in model dicot plant Arabidopsis. So, Arabidopsis during early stage of the development, they have a cotyledon type of leaf which are very small. During vegetative growth, they make rosette leaves, and rosette leaves even you can see a variation in the shape, size and morphology of the rosette leaf. And then you have a colloid leaves here. These kind of leaves are usually produced in the inflorescence stem after transition. Typical morphology of a leaf. So, in leaf you have a petiole here which is called leaf petiole, and this region is called leaf blade. Then as I said that you will have a midrib here, and then adaxial as well as abaxial sides. On one side you can clearly see that these kind of trichomes are developed, and then you have stomata formation, and this is a cross section of the leaf if you like. So, you have adaxial side, then you have palisade cells, then you have in between the vascular tissues, spongy cells, and then you have the abaxial sides. During the development, what happens that the pramodia or the program which is specific for the leaf development is getting positioned at the site of the leaf formation, and then during the differentiation these tissues are patterning, these are getting patterned, they are getting positions, they are taking a spatial identity, and then eventually the growth of a leaf is basically restricted. So, these are the some genes which is known during leaf development. So, Ushel and Clavata3 they are more kind of responsible for meristem maintenance. Then you have COC2. COC2 is, as I said, that COC1 and COC2 they are the boundary genes, they express very specifically at the boundary of shoot apical meristem and leaf pramodia. So, they are basically maintaining the boundary. Then you have STM and NOX gene which is regulating the meristematic activity. You have some other genes like asymmetric one which is regulating the leaf patterning, and then you have Yavi, filamentous and Kanadi as the genes which is regulating abaxial polarity. Fabulosa and other Fantastica is the gene which is regulating adaxial polarity. So, these genes are getting expressed and then they are regulating the function. Another important thing is that you have plenty of mutations. So, you have a genes which is regulating leaf morphology, and if you have mutation in that, you are going to see the defect in the leaf morphology. For example, if you look this mutant which is serrate mutant, so wild type, this is the morphology of the leaf, you have petiole, then you have the blade, but if you look here, there is a serration, there is a kind of change in the morphology of the leaf. Similarly, if you look asymmetric 2 mutants, you have a kind of similar kind of phenotype, but phenotypic severity is not very high. But if you make a double mutant between AS2 and serrate or AS1 and serrate, you can see that phenotype is severe. Similarly, if you take some of this Knox gene and if you ectopically overexpress, you can see that leaf morphology is totally disturbed. Another things what defines the leaf shape and size is basically curvature, or leaf curvature is one important parameter of the leaf. So, basically how curvature is maintained. So, if you have a cell, this is a kind of example, take a cell, and if the growth occurs uniformly, then you have a cell growth in a more kind of spherical manner. But if what happens that if growth dominates, if the growth in the central region is more than the peripheral region, then what you have, you get a kind of positive curvature, and you can see a structure like this or curve like this. On the other hand, if growth is more in the peripheral region than the central region, then you will have a kind of negative curvature, and then you can have this structure. Mutants of antirrhinum cincinnatus genes has been identified. Cincinnatus gene is a kind of class of transcription factor of TCP family, and what it was found that if you look this mutant, the morphology of leaf is extremely defective. So, this is a typical antirrhinum leaf, you have a petiole and then you have the structure and there is a proper shape, but in Cincinnatus mutants what you see that the curvature is totally disturbed in this mutant background. If you look the leaf arrangement here, if you look the defect in the leaf, so you can clearly see that maybe at the early stage difference is not very prominent, but at the later stage if you look this leaf, if you spread this curved leaf, you see this kind of structure. If you look the margin of this leaf and if you see the cellular feature of this leaf, you can clearly see that the wild type leaf and the Cincinnatus mutants leaf as a very different feature of the margin cells. Another genes which are known to regulate leaf morphology is Entacomata. If you look here, this is a typical wild type Arabidopsis root, but if you have loss of function of ANT, you can see that size as well as shape is somewhat similar, but size of this leaf is totally disturbed. And if you look, this is a wild type pattern. If you have loss of function of ANT, you see this defect. If you have gain of function of ANT, if you have overexpressed, you can see the opposite effect here. So, this suggests that ANT is also regulating leaf morphology. So, in general, if you look the development of leaf, so what happened in the first step in the shoot apical meristem or at the peripheral region of the shoot apical meristem, the first leaf primordia is positioned, and then this leaf primordia start distal growth which occurs in this direction. This generates a kind of two end, one is the proximal and the distal region. And then at third stage the blade initiates, where what happens, the marginal region or this region is basically getting developed into the blades and the basal regions are basically developed into the petiole. Later on there is a intercalary growth, it starts in the blade region, and that basically helps in the leaf blade to take a proper structure. This is the same schematic diagram or picture where you can clearly see a developing primordia. This is early primordia, this is slightly later primordia, but this is the leaf primordia which is at the stage of coming out. And there are some genes which has been identified to regulate the process. So, in the meristem, you know the meristematic genes are there which regulates the meristem function, but in the primordia you have other genes which are getting activated. One is that auxin label, and then auxin, what happens that it inhibits the meristematic gene. So, basically in the primordia region, meristematic genes need to be suppressed. The genes which are basically negatively regulating the differentiation program need to be suppressed. At the same time, the genes which are positively regulating the leaf specific developmental program, they need to be activated. So, this is what happens here. And another thing what I mentioned is that alloboid kind of genes are the genes which are present at the boundary between meristem and the leaf primordia, and they are very important to defining the zone or the spatially positioning the meristem or separating meristem with the lateral organ primordia. At the same time, you have a set of genes which are getting activated in the developing leaf primordia, and these genes are important for regulating leaf specific differentiation program. So, after this there is a process of differentiation. It can be either basipetal, acropetal or bidirectional, which means that differentiation can start from here and proceed in this direction, or it can start from the basal region and proceeds in the upper direction, or it can start from the both direction, and then it generates a kind of gradient. But once this gradient is totally so when both the ends are completed, then you have no gradient. When there is no gradient, essentially you can say that here the differentiation or growth of the leaves stops. So, if you look Arabidopsis thaliana, so in Arabidopsis thaliana it occurs in the basipetal manner. So, if you look, this is Arabidopsis leaf at different days. This is day 4, day 8, day 12 and day 16, and this is a cyclin promoter driving Gus. So, cyclin is a gene which basically marks the dividing cell. It is a cell cycle gene, so it is marks the dividing cell. So, if you look at the very early leaf, which is around 4 day 8, you can see the division occurring almost entire region of the leaf. So, this suggests that region is the differentiation is only restricted at the very tip of the leaf, whereas most of the leaves they are having cell division or proliferation activity going on. But once this leaf becomes older, so if you look in the 8-day-old leaf, what happens that the differentiation signal is migrating towards the downside. So, if you have now around 50% of the tip region, they do not have cycling activity. So, they are not showing the division activity. So, which means that the division activity is mostly restricted to the basal half region of the leaf, whereas the apical half region or the distal half region of the leaf, basically they lack the division activity, which means that they are undergoing the process of differentiation. Then if you see at more later stage like 12 days, the division activity is very restricted only in the very basal region of the leaf, whereas most of the leaf distal region of the leaves are showing pattern of differentiation. Slightly more later, if you look 16 days old plant, the entire leaf they lack the cell division or cell growth activity, which suggests that now this leaf is fully mature, it is fully differentiated. Now it will stop the property of differentiation. And then some of the genes which has been identified which regulates this property. So, if you look, if you divide this leaf, so there is a apical and basal or distal and proximal growth of the leaf, then you have this marginal growth, and you can see that some of the microRNA, some of the growth regulating factors, GRF are the growth regulating factors, and then you have Cincinnatus, TCP genes. The activity of these genes basically ensures proper shape, proper size, proper tissue patterning during leaf development. On the other hand, if you look the activity of YABI, homeodomain transcription factor, Canard genes, they basically ensures the adaxial versus abaxial property or polarity of the leaf. Auxin is also known to regulate basically leaf flattening in Arabidopsis. So, the auxin basically activates kind of or regulates the activity of auxin response factors. So, these are auxin response factor 2 to 4. Then you have MP, which is again auxin response factor 5. then some of the WOX genes, and their activity together basically ensures not only the leaf differentiation or leaf initiation, but also it regulates leaf flattening in this direction. Now, we will look the leaf polarity. So, as I said that leaf polarity is also very important, and because some of the features which are present in the adaxial surface is not present on the abaxial surface, and this polarity is very important, and polarity is regulated by a clear two category of the genes. So, one category of the genes which are specifically expressed at the adaxial site of the lateral organs, another category is expressed specifically on the abaxial site of the development. So, for example, if you look this growing pattern, and this is the messenger RNA distribution of FIL gene, which is abaxial gene. So, if you look the pattern, so this is developing leaf, you can clearly see that expression is mostly restricted the abaxial half of the leaf, whereas the adaxial half portion of the leaf do not have this expression, and this expression pattern is established very early even at the primordia stage. So, this is your meristem, and this is your first primordia here coming. So, if you look, if this is the primordia, this region total is the primordia, but expression is only restricted to the abaxial half of the primordia, but half adaxial primordia it totally does not express this abaxial genes. Whereas, on the other hand, or in contrast to this, if you look the adaxial genes, which is like revoluta or fabulosa, you can see it is showing a very opposite pattern. So, if this is a growing leaf primordia, you can see that the half primordia which is towards the adaxial surface is expressed these genes very high, but the abaxial side of the primordia it does not have the expression. So, the asymmetric expression pattern or transcript distribution or transcript expression of these genes essentially provides a proper polarity in the developing leaf. Apart from the homeodomain containing transcription factor, in abaxial you have Yabi domain transcription factor and Canardi some of the other genes. And what happens in this mutant background? So, if you have a mutant, for example, if you have a mutant in revoluta, what you can see that loss of function phenotype is totally where adaxial surface is totally abaxialized. So, you can see some of the picture here. So, this is wild type Antirrhinum leaves, but if you have a Fantastica mutant, Fantastica is a regulator of adaxial surface. So, if you do not have this gene expressed, you can see that these polarity in the leaves are totally lost. So, adaxial surface and abaxial surface both looks quite similar. And if you make a cross section, so as I say this is a leaf cross section. So, this is your adaxial side, this is abaxial sides, and you can see that xylem are usually positioned towards the adaxial sides, phloems are positioned towards the abaxial sides. But if you take the mutant and make a cross section, so it looks more kind of radial pattern, and here you can look that phloem is basically everywhere and xylem is in the center. This suggests that when you do not have adaxial specific genes, the all tissues are getting abaxialized. So, you have abaxial type of pattern in the leaf. On the other hand, if you have fabulosa dominant mutant, so fabulosa is again regulator of adaxial surface, it provides adaxial surface, but here it is a gain of function mutant. So, if you have gain of function mutants, then what happens that all the regions are getting adaxialized. So, basically even abaxial regions get converted into adaxial region. And if you look here the vascular pattern, here the vascular pattern is very different, it is opposite of the loss of function here. So, you can see here xylem is surrounding the phloem, which means that xylem is basically adaxial side. So, in this mutant background you can see that leaf is mostly adaxialized, whereas in loss of function the leaf is mostly abaxialized. If you look the cellular feature of the adaxial versus abaxial surface, so if you look scanning electron micrograph picture here, so in wild type adaxial surface looks like this, abaxial looks like this. So, in adaxial surface you can see is that there is a bulges of the cells, you can clearly see it is, whereas if you look the abaxial surface it is relatively smooth. So, bulges are not very strong, but if you overexpress ectopic overexpress filamentous or Yabi genes. So, remember filamentous and Yabi genes are regulated of abaxial surface. So, they basically promote abaxial surface. What happens, and if you look their adaxial surface, so adaxial surface normally in wild type looks like this. So, you have lot of bulges, but when you overexpress or you put filamentous or Yabi3, they becomes more similar to the abaxial surface. So, you can see they are becoming more kind of smooth wise. So, this suggests that when you have gain of function activity of Yabi3 or filamentous, basically they are converting adaxial identity to abaxial identity. But if you look the loss of function, so if you have a double mutant of Fil and Yabi3, so basically here both the abaxial promoting genes are lacking. In that case, if you look the abaxial surface, even abaxial surface look very similar to the adaxial surface. So, basically this is important. So, you have clear two category of genes, the adaxial promoting genes and abaxial promoting genes. If you do not have adaxial promoting genes, adaxial sites get converted into abaxial sites. If you do not have abaxial promoting gene, abaxial surface get converted into adaxial surface. And they are actually regulating their activity as well. So, if you look here, so this is the expression of Fil, which is abaxial gene, and it is position, it is mostly expressed in the abaxial side of the leaf and it is absent in the adaxial side. But if you have gain of function mutant of fabulosa, basically what happens, the gain of function mutant of fabulosa means every side is adaxial sites, and this if you look the expression pattern of now filamentous gene, because filamentous is abaxial, now in these mutants you have everything adaxial, then you can clearly see that if you have a homozygous gain of function mutants of fabulosa 1D, you can see the expression is almost disappeared. So, there is no expression of abaxial specific genes. On the other hand, if you look here, in some of the cases the expression is there, but it is very restricted, it is not the expanded. So, basically the domain of expression is highly restricted. And if you look some of the organs which are basically adaxialized, you can clearly see that their expression is very low and extremely disturbed expression pattern. On the other hand, if you look the expression pattern of adaxial promoting gene in the abaxial mutant. So, Canaday genes are basically responsible for promoting abaxial side. Now, if you have double mutant here and if you look now revolota, fabulota, these two genes are adaxial side genes, you can clearly see in mutants background their pattern is totally disturbed. So, if you look in the wild type, they are clearly localized towards the adaxial surface, but if you look the mutant here, you can see they are quite uniform, or you can see even expression in the abaxial surface. And similarly, if you have Canaday mutants, the abaxial genes they have lost their expression or they are very low level expressed. So, if we summarize, if you look first the growing leaf, you have the meristematic zone and then you have the primordia, and then in this primordia the adaxial versus abaxial polarity is already established, and in adaxial side what happens that these homeodomain containing transcription factor, which is fabulota, revolota, fabulosa, they are basically active in the adaxial surface. On the other hand, Canaday, Yabi, these genes are active on the abaxial surface and they mutually inhibit each others in their domain. So, in adaxial domain these genes inhibit expression of Yabi, in abaxial domain these genes inhibit expression of homeodomain genes, and this kind of mutual exclusive expression pattern of adaxial promoting genes and abaxial promoting genes basically establish the adaxial versus abaxial polarity. On the other hand, there are some of the microRNAs, trans-siRNAs, they are also playing a very important role in establishing and regulating polarity in these organs. If you look the cross section of the leaf, so you can clearly see that you can have a two domain, the adaxial domain and the abaxial domain. So, these are the adaxial domain. In adaxial domain, adaxial genes like these they are basically active, and apart from the adexal genes you can see that genes which is responsible for the promoting leaf identity, they are also getting expressed. On the abaxial side you can have some of the abaxial promoting genes which are getting active. Apart from that you have the activity of auxin, microRNA, auxin response factors, MP, and some of the other key regulators of leaf differentiation. So, all together activity of hormones, activity of transcription factors, the coordination between meristematic activity and the leaf primordia set complex genetic program which is regulating proper patterning, proper differentiation and proper organ development in plants. So, I will stop here. In next class we will look floral transition and flower development. Thank you very much.